my pleasure to introduce Dr. Dolan. She is currently an assistant professor at Johns Hopkins University in the Department of Neuroscience, and I believe she's a rising star, if not already a star, in the neurobiology of social behavior and social cognition. She received her undergraduate training at Duke University and then obtained her MD PhD at Brown University and MIT, where she studied Fragile X syndrome in the laboratory, and that is one of the most common identified forms of autism, and also saw patients with autism at Massachusetts General Hospital in their genetics clinic. Her work identified targets for the treatment of autism that were then pursued by two different pharmaceutical companies, Seaside Therapeutics and Pfizer, for whom she served as consultants. She also continued her training in one of the leading molecular neuroscience laboratories in the world, that of Dr. Robert Malenka at Stanford University. And Dr. Malenka wisely gave Gül the freedom to lead a project looking at the neurobiological mechanisms of social reward. And that's where I got super excited, because she discovered that serotonin 1B receptors in the ventral striatum play a critical role in social reward, which is my own area of research. So it's pretty exciting for me. Gül has been at Hopkins for just a little over a year, and during that time alone, she has received the 2015 Cero Scholarship for Exceptional Young Scientists, and this is a competition of 186 assistant professors from 126 countries. She received a Biomedical Research Award from the Hartwell Foundation, and she's been awarded 2 NIH grants. Her research on autism is published in the most prestigious journals and is truly groundbreaking. And so I'm very excited that she is taking the time to be with us today. So please join me in welcoming Dr. Gül Dölen.
Is this on? Yeah? Okay. Thank you, Janet. And just so there's no mistaking, I actually don't have 2 NIH grants. I wish. The Hartwell and the Searle are my 2 grants. But, here's hoping. But thank you for that really wonderful introduction. And thank you all for being here. I haven't been to Arizona since I was maybe 12. And I'm just so amazed by the weather here always. I really love the dry heat and what it does for my hair. And it's just so beautiful out here. You guys are really, really lucky. So today I'm not really going to be telling you anything about what we've been working on in my own lab, because most of the data that we've generated, which is a very small amount of data, is just not ready to be talked about. So I'm going to tell you about work that I did as a postdoc that Janet alluded to in Rob Malenka's lab. And I'm not really going to tell you so much about the autism pathogenesis part. I'll sort of touch on it at the end. And that was work mostly that I started in Mark Baer's lab. And combining this social reward and autism pathogenesis is really the direction that my own lab is going to be taking, so stay tuned. So when most people think about social behaviors, I think the 2 most common that people think about are interactions between people who are in love, your spouse or your boyfriend or your girlfriend, or the attachments that people have to their children. And I think part of the reason that those are very prominent in our mind is incredible work that's been done in the prairie voles that have really tried to work out mechanisms for how those behaviors are encoded by the brain. But there's another type of social interaction, and it's one that's really, really important to humans, and it's the one that we're having, for example, right now. And so whatever nervousness I feel for being up here and speaking to all of you is a little bit, the fear of total social rejection by this whole group. And, whatever excitement I feel about being here is that, I might be able to convince you, some of you, to get excited about this kind of work and maybe want to collaborate and, really convince you of my ideas. So there's this other type of social interaction between our community members, our group members, that, is really important to us. And I think that Jerry Seinfeld actually has a little bit about this, which I think really kind of turns, Maslow's hierarchy of, needs, which, kind of puts food and water at the very bottom, and then shelter and social somewhere in the middle, and then, achievement and all that is at the top. And I really think that this, maybe makes us wonder, should social be moved down to one of the more primary needs? And I'll just show you this little clip. Oh, I don't have volume. Let's see, I think I can do it. Maybe here. That's okay. Let's see if I can just, okay. All right, I think we're good now. So we'll start again.
I saw a thing, actually, a study that said speaking in front of a crowd is considered the #1 fear of the average. I found that. Number 2 was death. Death is number 2? This means to the average person, if you have to be in a funeral, you would rather be in the casket than doing the
Yeah, so I think this is a sort of funny way of illustrating how important social interactions really are to us. But beyond that, social interactions, seem to be really important for our longevity. So, there have been a lot of studies out there that have shown that social network size and the strength of your social community is really important for your general health. But what I love about this study, published a few years ago, is that this was sort of a meta-analysis of a bunch of different risk factors that are known to affect longevity. And it sort of put them all together and compared the effect size against all of these. And so, somewhat surprisingly, the effect size of air pollution and hypertension and obesity and exercise and being vaccinated and smoking and alcohol all are, less than the importance of social relationships. So this is a really important part, and we don't really understand why that is, why these are so critical to our longevity. And there are a lot of people who are interested in this. But I like to show this just in case you were wondering if this type of reward that I'm going to be telling you about isn't as important as food or water. The other reason that I'm really interested in social interactions is because this disease, autism, which was first described by Lee O'Connor in 1943, actually at Johns Hopkins, and he described it as a profound lack of interest in others. And today's definition includes impaired social and communication behaviors, and then stereotypies and intense idiosyncratic interests. But this social interaction deficit is complex. So this is a picture of a patient with Fragile X that was given to me by one of the parents at a parent group for Fragile X patients. And what you can see is that this kid is at the party. He's near the other kids, but somehow there's enough social anxiety associated with looking at the camera that he has to cover his eyes, and he's not really in the mix with the rest of them. And so disentangling what causes these social impairments in patients with autism, I think, is going to require a more sophisticated understanding of how the brain encodes social interactions. And I think the simplest hypothesis about this profound lack of interest in others would be that social interactions are just not rewarding for patients with autism. And so we wanted to kind of explore some circuits and functional mechanisms for social reward in order to put ourselves in a position to test that hypothesis in autism. Social behaviors are common in the animal kingdom. They're very varied, so it ranges from trophallaxis, which is food sharing in bats, to the sort of altruistic behaviors that we as humans do, and pair bonding in prairie voles. And this one that I really love, this is a singing squirrel who basically vocalizes to alert his conspecifics about the approach of a predator. And what's so interesting about this social behavior is that this animal is putting himself at risk in order to help his larger community. And because of this conflict between what's good for the individual and what's good for the group, a lot of people have suggested that, social reward might be that this social interaction must be rewarded by the brain in order to have this type of behavior persist. And so some of the early work that I mentioned a few minutes ago about the mechanisms that might encode this social behavior came from work in prairie voles and mountain voles and mice, comparative studies. And what these studies showed is that in this species called prairie voles that exhibit this pair bonding behavior where they mate for life and both the male and the female take care of the young, but not in mountain voles and mice which don't do this behavior. They saw an increase in the expression of oxytocin receptors in this brain region called the nucleus accumbens. And as you can see in mountain voles and mice, this upregulation of receptors was not impaired. And this is done by receptor autoradiography. So the nucleus accumbens is part of the mesocorticolimbic reward circuit, which canonically involves the VTA dopamine neurons, which send projections to both the nucleus accumbens and the prefrontal cortex. The accumbens receives projections from the prefrontal cortex and other brain regions, and then sends it along either directly back to the VTA or with an intermediary through the ventral pallidum. And this reward circuit has long been implicated in drugs of abuse and addiction and other rewarding behaviors. So the fact that oxytocin receptors were upregulated in the accumbens of prairie voles is very suggestive that oxytocin was working to enable social attachments seen in pair bonding. Even though mice don't do pair bonding behaviors, they are very social. In the wild, they live in these groups of 5 to 10 animals called demes, which are adult animals living together. They have alloparenting responsibilities. They share territorial defense responsibilities. There's some evidence that they engage in social play and empathy and also vocalizations. And these are sort of the hallmarks of sociality. And so they are very social. And yet the oxytocin receptor expression in the nucleus accumbens, according to these earlier studies, really sort of left us without a mechanism for how these other types of social interactions might be rewarding. So we wanted to test this, and a couple years before we published this work, there was a paper by Panksepp and Lavis. I don't know actually how to pronounce that, but that's how I've been pronouncing it, Lavis. And they had described this social condition place preference behavior. And this behavior basically, condition place preference is something that's used in the reward literature commonly, and it's often used to look at cocaine reward or the rewarding properties of many drugs of abuse. And they adapted it to look at social reward. And so we adapted this behavior to be a little bit shorter for technical reasons that I'm happy to discuss if you're interested. And so the adapted way of this, I'm just going to go through this because I'm going to use it throughout the rest of the talk, is the animals start off in their home cage with their cage mates, who are usually litter mates. And then during the preconditioning trial, they go into an activity chamber that has 2 different types of bedding separated by a barrier that they have free access between the two compartments. And then we give them 30 minutes to sort of run around and explore this, and we measure how much time they spend in each of the 2 types of beddings as a baseline. And then they're moved to a new home cage with bedding, which is one of these 2 types that they've seen for the first time in the preconditioning trial. And they live there for 24 hours with their cage mates. After 24 hours of social conditioning, they're then moved to the other type of bedding, and now they spend 24 hours by themselves in the other type of bedding. And on the post-conditioning trial, we ask how much time they spent on the bedding that was conditioned by themselves versus socially. And social conditioned place preference is evidenced by spending more time on the bedding that was conditioned to the social. And what we found is that this behavior, we were able to show that with IP saline injections, these are the individual responses of the animals, and then the average responses. 900 seconds indicates that they spent equal amount of time on the two types of bedding, and that with saline, the animals show a nice increase in the amount of, a significant increase in the amount of time they spend on the social bedding in the post-conditioning trial. When we do IP oxytocin receptor antagonist injections, however, we don't see this social conditioned place preference in the post-trial. And these are just normalized and subtracted preference scores between groups just to show, to compare across these groups. Next, we wanted to know whether or not oxytocin was acting in the nucleus accumbens to enable this reward, this social reward. And so, sorry, this is mislabeled. This is not IP saline and IP oxytocin receptor antagonist. This is now nucleus accumbens injected. Oh, no, sorry, I'm sorry. Just skip that slide. Sorry. We took advantage of this Andelman probe that had been developed in Mike Fee's lab to do microdialysis in reverse microdialysis in the awake bird. And we love this technique because it allowed us to get drug in a semi-continual manner into the nucleus accumbens without having to anesthetize the animals. And so we adapted this for the mouse, and then we were able to put in this reservoir either saline or oxytocin receptor antagonist. And what we saw is that when we did saline, we got nice social conditioned place preference, but oxytocin receptor antagonists in the nucleus accumbens blocked this social conditioned place preference. So next we wanted to know, well, what would be a potential source of oxytocin in the nucleus accumbens? And so work from the Perivol has suggested that there were these magnocellular neurons that send projections to the posterior pituitary and release oxytocin synaptically, and also somatodendritically released oxytocin into the CSF. And these magnocellular neurons are thought to be releasing somatodendritically because their expression of oxytocin receptors in the perivols goes up, even though there isn't an increase in the number of axonal projections to the nucleus accumbens in that brain region. And so this mismatch between axon projections and receptor expression has led people to believe that there's a somatodendritic release mechanism. In addition, there are these other neurons called the parvocellular oxytocin-ergic neurons, and we wanted to see if there was any evidence for a parvocellular input directly to the nucleus accumbens. And this is just to show you that when people have looked across species, there are oxytocin antibody expressing projections that are running through the accumbens across species, including mice. But we wanted to look at a more gold standard approach because these kinds of studies can't distinguish between axons that are just passing through or ones that are actually making synapses. So we took advantage of this not so new, but newish technology that was developed by a number of different labs, but then also perfected in Rob's lab, which is the rabies virus. So this rabies virus has now been genetically engineered. So the glycoprotein that is normally encoded by the rabies virus is replaced with whatever we want, in this case the fluorofluor GFP. And then this rabies virus that doesn't have the glycoprotein and instead has the GFP isn't able to get into presynaptic terminals because it doesn't have the glycoprotein that's recognized by the terminal. And so we grow these viruses up in, okay, this died, so I'll just tell you. We grow them up in BHK cells that are expressing the glycoprotein on their surface, so when they bud out, they incorporate the glycoprotein. And what that means is that these oh, thank you. Okay. That that these rabies virus now are able to get into the presynaptic terminal, but then because they don't make the glycoprotein themselves, they can't jump to the next synapses. So they go to one presynaptic input and then stop. Okay? And so using this technology, we injected the rabies virus into the nucleus accumbens, and then we asked, do cells that are sending exons to the nucleus accumbens from the paraventricular nucleus also express oxytocin? And so we saw evidence for co-localization between the oxytocin antibody and the rabies virus expressing neurons that are projecting to the accumbens. And in general, these were the diameter and the anatomical location of these suggested that they were parvocellular neurons. What's more is that when we did this same manipulation and now look at the supraoptic nucleus where the magnocellular neurons are exclusively located, so there are no parvocellular neurons in the supraoptic nucleus, then we did see evidence for some cells that were sending projections to the accumbens, but these were not oxytocinergic. Oops, sorry, I showed you the wrong thing. And what this suggests is that the difference between prairie voles and rats and mice is not just that they have a difference in the expression of oxytocin receptors, but also that the magnocellular projection from the SON is absent in rats and mice and present in prairie voles, suggesting that it's really this magnocellular input that's responsible for the pair bonding. Because we're electrophysiologists, we next wanted to know whether or not we could detect a synaptic effect of oxytocin in the nucleus accumbens. In the nucleus accumbens, 95% of the neurons are medium spiny neurons. They're GABAergic neurons, and they send projections to the ventral pallidum and the VTA. And these cells receive inputs from glutamatergic inputs from the prefrontal cortex as well as a number of other regions. And so we did whole cell patch clamp recording in these medium spiny neurons and asked what were the excitatory responses and what did oxytocin do to those responses. And so when we did these recordings, we collected a baseline and then bath applied oxytocin, which induced a robust about 20% decrease in the amplitude of the EPSC, which was blocked in the presence of the oxytocin receptor antagonist. And this is just summary data showing those comparisons. Furthermore, because oxytocin is a peptide, we wanted to rule out the possibility that what we were seeing as a decrease in EPSC amplitude wasn't in fact just a receptor occupancy issue. So then we did these washout experiments. We washed out oxytocin, and then at some interval later, we added in oxytocin receptor antagonist and to see if this would be reversed, and it wasn't. So this really suggested that the oxytocin is inducing a true synaptic plasticity and rather than having a decrease in the amplitude that's due to receptor occupancy. We also wanted to be able to tie this synaptic effect back to the rearing history of the animal. So we compared the magnitude of this oxytocin LTD in animals that were reared normally under social conditions or isolated for 1 to 3 days. And in this case, what we saw is that the animals that were isolated had a larger magnitude of oxytocin LTD compared to the animals that were socially reared. And the interpretation that we like for this is that this suggests that the social experience is actually occluding the oxytocin LTD, although there are other explanations which I'm happy to discuss. Okay, so we had an effect of oxytocin, but now we needed to find the receptors. There aren't any specific antibodies that can recognize the oxytocin receptor differentially from the vasopressin receptor. So people have used this reporter mouse, which is the oxytocin receptor venous knock-in mouse. And the important thing to note about this strategy is that what this tells you is which cells are making the oxytocin receptor but not necessarily where those receptors are localized, which is important in the case of presynaptic localization of receptors, as you'll see in a minute, because axon terminals can be very far away from where the cell bodies are. So when we just looked at the cell bodies of different cell types within a nucleus accumbens, so DARP to label medium spiny neurons, chat cells, and NOS, we didn't see any co-localization between the oxytocin receptor expressing neurons and the cell type specific antibody. We did see a few cells that were PARV positive and oxytocin receptor expressing, as well as glial cells expressing GFAP. So the next experiment was to ask whether or not, if we knocked out oxytocin receptor in those cell types, what effect would that have on social CPP? So now we used a different type of virus, the adeno-associated virus, and we used the DJ serotype that was discovered by Mark Kay at Stanford. And this virus has the property that when we inject it into the nucleus accumbens, it will just go into the cells that are local, and it doesn't go retrograde or anything. It's just locally wherever we injected it. And we had this adeno-associated virus now expressing CRE, so that we could knock out the oxytocin receptor in these conditional oxytocin receptor knockout mice in the presence of CRE. And when we did this manipulation, what we found was that the social CPP was normal in both wild type and conditional oxytocin receptor knockout mice, suggesting that those oxytocin receptors found in the nucleus accumbens that I showed you before were not affecting social CPP in any measurable way. Next, we wanted to look to see whether or not the effects of oxytocin were presynaptic or postsynaptic. So electrophysiologically, we can determine this by looking at mini EPSCs, and we can look at the frequency and amplitude of these mini EPSCs. And what we saw is that the oxytocin induces a change in the frequency, but not the amplitude. Furthermore, the paired pulse ratio of EPSCs was increased after oxytocin, as was the coefficient of variance. And all of these things taken together really suggest that oxytocin is acting presynaptically. So the next question is where? So we know that they're presynaptically active, but which presynaptic receptors? So then we used our rabies virus strategy again, and this time we used rabies virus expressing TD tomato, and we injected them into the oxytocin receptor venous mice. And we looked at all of these different brain regions. I'm not going to go through them in detail, that project to the nucleus accumbens and see whether or not there was co-localization as evidence for a putative source of presynaptic oxytocin receptors. And so we looked in the prefrontal cortex and the olfactory nucleus and the hypothalamus and thalamus and amygdala and dorsal raphe and VTA and the ventral subiculum of the hippocampus. And many of those were putative sources. So in order to test whether these putative sources of oxytocin receptors would have any functional consequence, we now used rabies virus expressing CRE in the conditional knockout mice. And when we did this manipulation in wild type mice, we got beautiful social CPP, but in the conditional mice, we got interfered with the social CPP. And this is just comparison. So we had all of these putative locations, and we didn't know where. And I left this deliberately messy and sort of overwhelming because I sort of wanted to impress upon you what a big challenge it is when you find a plasticity that's presynaptically mediated, especially in an area like the nucleus accumbens, which receives inputs from so many different areas. And so the way that we attacked this problem was really by brute force. We just went around to different brain regions that had been implicated by our rabies virus study and did AAV CRE injections. And I'm not going to show you all the negative data. I'll just jump to to the dorsal raphe, which worked. And so the AAV Cree injected directly into the dorsal raphe interfered with the social CPP, whereas in the conditional mice, but not the wild type mice. And so this really suggested that, This really suggested that it was oxytocin receptors on the dorsal raphe inputs to the nucleus accumbens that had the oxytocin receptor. And since the dorsal raphe contains a lot of different cells, not just serotonin, we wanted to make sure that it was the serotonergic neurons that were projecting to the nucleus accumbens that were expressing the oxytocin receptor. So now we did our rabies virus trick again, and then we counterstained with the tryptophan hydroxylase to label serotonergic neurons. And what we found is that nucleus accumbens projecting serotonergic neurons express the oxytocin receptor, as shown by co-localization. Gosh, I don't know what happened here. We're getting some repeats. Sorry. I'm sorry. I don't know what's happening. Oh, okay. So David Lovinger's lab had shown, actually, luckily for us, that serotonin bath applied to the nucleus accumbens would induce a robust long-term depression. And they had characterized the serotonin LTD and found that it was mediated by the serotonin 1B receptor, which is abundantly expressed in the whole striatum, and including the ventral striatum, the nucleus accumbens. And so we were able to replicate that using the serotonin 1B agonist. And this agonist induced a robust LTD, which was not there was no extra LTD when we bath applied oxytocin. And so what this really told us is that the oxytocin LTD and the serotonin LTD might be in the same pathway.
Can I ask a quick question?
Yeah.
I'm sorry if I forget. What's the presynaptic stimulation in these experiments?
We're stimulating just using a regular metal electrode, and we're recording EPSCs because of the configuration of the drug.
Stimulating just within the nucleus accumbens itself?
Yes,
Okay.
and so we make the argument that we're preferentially getting the prefrontal cortical inputs because we've cut sagittal sections and we try and put the electrode rostrally, but really in the nucleus accumbens, the inputs kind of come from everywhere, and so that's sort of a soft argument that we used to use before optogenetics. And so if we really want to know which inputs we're targeting, we have to inject channel rhodopsin into all of the input regions and then shine light. That way we can really know that we're looking at, prefrontal cortical versus thalamic. But in general, we think we're biasing ourselves towards prefrontal cortical inputs, but that's a caveat that hopefully in my own lab we'll be able to get around. So the fact that oxytocin does not induce a further change in the magnitude of the serotonin 1B LTD suggests that these two transmitters might be in the same pathway. In order to test that formally, we asked whether or not oxytocin LTD could be blocked by the serotonin 1B antagonist. And indeed, when we do induce the oxytocin LTD in the presence of the serotonin 1B antagonist, it's significantly blocked. And that leads to the strong prediction that if we microdialyze the serotonin 1B antagonists into the nucleus accumbens, that we should block social CPP. And indeed, when we do saline injections into the accumbens, we get normal social CPP. But when we do serotonin 1B antagonists into the nucleus accumbens, it's blocked. And so this leads us to the following model, that PVN, parvocellular oxytocinergic inputs to the nucleus accumbens, are releasing oxytocin, which is binding to oxytocin receptors on serotonergic terminals in the nucleus accumbens. and that the activation of these oxytocin receptors leads to the release of serotonin into the synaptic area, and that these are seeing serotonin 1B receptors on glutamatergic inputs to the accumbens, and that activation of serotonin 1B receptors is leading to a change in the probability of release in the glutamatergic inputs, which we are recording in the medium spiny neurons as LTD. We think that the reason that this mechanism had been missed before is because I sort of hinted at it at the beginning, because we don't have a receptor antibody, we had been forced to use receptor autoradiography and transcript tagging to identify neurons. And these might this might make it difficult to detect receptors that are G protein coupled, since G protein coupled signaling is thought to be important for amplifying a signal. So a very small amount of receptors could nevertheless have major functional consequences. And so this technique doesn't have the sensitivity to detect those small numbers. And as I mentioned before, transcript tagging doesn't tell you where the receptor is expressed, it just tells you which cells are making the receptors. And since these are presynaptic, this technique wouldn't be able to detect them. Furthermore, we think that because social living evolved probably 30 million years prior to pair living, that this mechanism that we're describing might be a predecessor of the evolutionary specialization seen in prairie voles. And furthermore, this type of social behavior that we're describing with social CPP in comparison to pair bonding might be the type of social interaction that is relevant to autism, as I mentioned before. And so using these tools, we're hoping that we can dissect the pathogenesis of autism. And so this is just a list of autism candidate genes from the Simons Foundation website. It's actually much longer than this now. This is a little bit outdated. We're up to somewhere on the order of 650 genes. And oxytocin receptors and serotonin 1B receptors are on this list. But the etiological diversity of this is very daunting. And so I think what we're going to need is better tools to understand the circuit level functions that are disrupted in autism so that we can make sense of this heterogeneity and try and come up with explanations or mechanisms for how, despite the heterogeneity, we can have a unified disease. And so with that, I just want to thank the members of my lab and Rob's lab, as well as the Sudoff lab. I didn't really show you any of this Fragile X work, but that was done in Mark Baer's lab. And I especially want to thank Aaron Andelman, who made those probes, and he's in Carl Dyserath's lab. So I think we have plenty of time for questions. Yeah.
Beautiful talk. I have two questions for you. So one was, you had that slide showing that GFAP positive astrocytes actually express oxytocin receptors.
Yeah.
And the follow work from Ben Barris and Comparative work from looking at synaptic changes. Do you think that has any role in changing some of the dynamics of the circuit? And then the second question really was just, I was wondering if you could comment on some of the oxytocin clinical trials that have gone on with autism and what your thoughts were on that broadly.
Sure. So for the GFAP, the glial cells, it's actually been known for a while that glial cells express oxytocin receptors, and people have looked at how they might be regulating oxytocin release, and we just didn't really focus on that, mostly because when we did the AAV pre-injections into the nucleus accumbens, we should have knocked out those GFAP positive oxytocin receptors. And it may have an effect, but it's too subtle to be detected by the social CPP assay that we're using. So we'll have to come up with more fine-grained behaviors, I think, to figure out what those glial cell oxytocin receptors are really doing. And I think we might get a better sense if we started with electrophysiology and then moved our way up to behavior. For the clinical trials, I actually just wrote a review article about this. And this is sort of a scary summary slide, kind of most of which I told you about. But what I actually think is that there are two parallel pathways of oxytocin-ergic signaling in the brain. So there's this magnocellular pathway and the parvocellular pathway. And the magnocellular pathway, I think, is really mediating the functions of oxytocin that have these sort of big effects that are not synapse-specific, but rather reflect the activity of oxytocin floating around in the CSF and being released systemically, and sort of coordinating central and systemic functions of oxytocin. Because it's known that oxytocin in the systemic circulation, can bind to receptors that are not just in the breasts and the uterus, but also all over the body, really. And so this type of magnocellular pathway activity of oxytocin, I think, I call it the mad love pathway. So it's, falling in love with somebody or falling in love with your child. And, this type of response generalizes. So when you're in love, everything is roses, and you cease to recognize the faults of your chosen mate, and you can put up with crying in the middle of the night and all of those kinds of things. And so I think that this sort of matches a paracrine type of function of oxytocin bathing the whole brain. In contrast, the kind of responses that I talked about today that are probably used to determine in-group, out-group membership and might require more cognitive decision-making about, trustworthiness, reliability, reciprocal altruism, that these might be more likely to be mediated by something that's, very temporally precise, and the amount of oxytocin that's being released by these medium-dense core vesicles is about 1/8 the amount that's released by these large dense core vesicles. Plus medium dense core vesicles, they're only, usually 1 or 2 per terminal, whereas large dense core vesicles, they're, somewhere around 100 or more of these can be at one of these magnocellular release sites. And so the temporal and spatial specificity of this, I think, matches more the type of cognitive social behavior. So now this is the long answer to your question about autism, but most of the oxytocin trials in autism, in fact, all but 2 that I know of that were, randomized double-blind controlled trials, have used intranasal oxytocin. And intranasal oxytocin has been very hotly debated whether or not it can even get into the central nervous system. But there's a little bit of evidence that, the nasal epithelium is a leaky blood-brain barrier because the cells are turning over so fast. And so the integrity of the blood-brain barrier might be less there. And so the idea is that oxytocin would be getting into the circulating CSF. And so assuming that it gets into the brain, I would think that the intranasal oxytocin is sort of mimicking this magnocellular type of release mechanism. And but also it's probably getting into the systemic circulation through the nasal capillary bed and acting at the release sites in the posterior pituitary. Oxytocin is known when it's released into the systemic circulation to have it binds to receptors in the adrenal cortex and regulates stress hormones. So I think that the positive effects that have been reported for the intranasal oxytocin trials in autism really reflect this sort of magnocellular release mechanism and sort of are addressing the problems in autism that are associated with social anxiety or hypersensitivity to sensory stimuli, but not so much the social cognition problems that I think are sort of the hallmark of classical autism. And so, because I think those types of cognitive, social cognition problems are mediated by this type of signaling.
What do the other two trials do?
They just did IV, so even more evidence that it's probably systemic. And another thing is that they haven't all been consistently positive. So the ones that have been negative have had larger numbers of patients, so 40 to 50. But the other thing is that the outcome measures used in the larger trials were some, sort of global measures, which are really bad. I mean, I personally think they're sort of useless because what they are things like the clinician-guided interview of, global impressions, and they're very subject to placebo effects. And when we compare to the types of measurements that we do in mice, which can be very, very sophisticated in looking at synaptic plasticity and dendritic spine changes, and we never look at two mice and we go, yeah, less autistic, And so we're comparing apples to oranges when you use a global measure compared to what we do in animals. And the ones that have, shown more positive effects have been more specific assays, like measuring differences between emotional and cognitive empathy, measuring amount of time that the person spends, gazing at a picture of someone's eyes. And I think the important thing is that if moving forward with clinical trials, we have to get more sophisticated about the type of endpoints that we use. And then we also have to have a better appreciation of sort of the subtypes of autism that might manifest as, social anxiety, but are really, or social cognition deficits that are really just a social anxiety deficit, because I think that they're going to respond very differently to not only different drugs, but different modes of administration. So I think we have to start to get a little bit more sophisticated in how we describe and talk about autism in the human patients. Yeah.
Has the oxytocin ever been combined with serotonergic drugs, and do you think there would be some benefit to the additive based on what you're finding?
I'm not sure if it would be additional effects, because I think that most of the oxytocin effect is being mediated by serotonin, but I'm not 100% sure about that, because when we have tried to optogenetically induce CPP by stimulating cell bodies in the dorsal raphe, just doing that by itself doesn't seem to produce a CPP. And so another lab that I've talked to about this, who's kind of done the same kind of experiments, Zach Neiman's lab, they also find the same thing. And what he suggested is that it's possible that the recruitment of serotonin by oxytocin doesn't actually require that the serotonergic neurons be firing, right? So it might be something special about that recruitment that's different from just serotonergic neurons firing in general. That being said, there are clinical trials of SSRIs in autism, and I think the problem there is that, and kind of the reason that I stayed away from serotonin for so long is that there are 14, maybe 15 subtypes of serotonin receptor, and right now all of the clinical trials that are looking at serotonergic interventions are treating it as if it's just one big lump sum. And there are known effects of serotonin 2A and 2B in the hypothalamus that might have opposite effects on oxytocin regulation in the hypothalamus. So I think we have to stop taking this sort of black box approach to transmitters and recognize that it's not just which one, but when and how and to which receptor.
Can you say more about what the social experiences were like that these mice had during the condition dose preference training? Were they with same sex or different sex? Were they fighting? Were they having sex? What was going on?
So these were, I did some pilot experiments with males and females, but most of these experiments were just males. Most of the time, you know, I don't want to say all, but most of the time, they were their litter mates, and they were animals that, you know, they were familiar with.
Sexually mature?
They were P30, so not quite. At P45 is when they become sexually mature. And just to give you, I mean, we're a little bit interested in this question because just to give you a sense, you know, there's a fair amount of variability in how much they seem to prefer the social side. And we think that this probably reflects something about the animal's position in the dominance hierarchy, because even though they're not, you know, sexually mature yet and really established strong dominance hierarchies, we think it's still possible that this kind of thing is starting to come online by P30. And so P30 to 35 really. And so we want to kind of...
Can you just put, that graph makes it look like there's about 5 mice that have a big difference. One went down dramatically and most of the rest did the same.
No, I mean, there's some overlapping situations. Let me see if I can find another one that you can see. Yeah, I mean, most of them kind of head in the right, in the preference direction. There are a few that go in the wrong direction, some that kind of stay the same. You know, there's a huge amount of variability, right? And so I think that exactly the kind of thing that you're asking about is at play to explain this variability. You know, I think that we kind of, in general, assume that these are pleasant social interactions, but if you're the low man and you're getting beat up, maybe you don't think they're so great. And so we want to start playing with all of those different parameters to see if we can make sense of some of this variability. Yeah.
Because the nucleus accumbens is so important, like the reward circuitry, do you think in autism maybe they're not finding these social reactions like rewarding? Has anyone ever looked at, or like is oxytocin affecting like the dopamine reward circuitry? Like maybe it's not rewarding, so like maybe, I don't know, kind of think like anhedonia, like they're just not getting reward out of this, or what I interact again?
Yeah, so I mean, one of the, I think the simplest hypothesis is that exactly that oxytocin is necessary for having a pleasurable feeling in being social, and that in autism, you're just not getting that social pleasure. It could also be that there's just some general effect on reward, and so it's not specific to social, but that, you know, they just don't respond to rewards at all. The human studies looking at this have been very confusing because, unfortunately, they have used monetary rewards. And so, you know, studies that have used monetary reward and looked at reward profiles in autistic patients have found that there's a generalized, you know, insensitivity to reward. But other studies who have used the rewards that, you know, the patients themselves, you know, like toys and cars and trains, you know, have found no generalized deficit and really suggest that the social, it's domain specific to social. And I didn't show you this data, but also we looked at whether or not oxytocin antagonists disrupted cocaine CPP, and it did nothing to the cocaine CPP. And so that's a little bit in contrast to Ian McGregor's work, because he really has suggested with a handful of papers that oxytocin might be important for cocaine and methamphetamine CPP. But I have to emphasize that in all of his studies, he was doing IP injections of oxytocin, and so that has two problems. One is the one that we've already talked about, but the second one is that I think that especially in the case of a peptide that has low fidelity for its own receptor and can, you know, bind also to the vasopressin receptor, that when you're talking about agonist studies, it's just not as informative as antagonist studies, especially because the antagonists have much, much higher selectivity for the receptors. So, I don't like those studies for those two reasons, and because I wasn't able to replicate them, I don't really believe them. So, yeah, I mean, I actually was sort of expecting that there was going to be some effect, because I was actually sort of thinking, well, maybe one of the reasons that cocaine is so rewarding is because, it makes you feel confident and Superman, and you can socially interact with anyone, and that it's got this social, and it does have a social component. People who use a lot of cocaine like to party and go and be social, and so I thought maybe it would have an effect, but I just didn't see it. And I think it may be the other way around, that actually, cocaine, it makes you more confident, so you're able to overcome whatever social anxiety there is, but that it's not necessarily social reward per se.
We have time for one, maybe two more questions.
Do you know if there's any change in the population of 1B receptors in autism? Like, has anyone looked at that compared to control?
I don't know that anybody has looked at that specifically. The study that looked at oxytocin 1B for, as an autism gene candidate was just looking at polymorphisms in patients. So I don't believe that they ever correlated it back to expression of receptors themselves.
I have another quick question.
Yep.
Is the human brain more like the variable or the non-variable in terms of its distribution of oxytocin receptors?
I think it's probably more like the prairie vole. However, Larry Young has looked at this a little bit. The evidence is actually a little bit stronger for vasopressin. So there is this polymorphism in the upstream of the UTR for both the oxytocin receptor and the vasopressin receptor, which kind of evolved from a gene duplication event. So it's not that surprising that they have the same kinds of polymorphisms. And this polymorphism has been associated with increased expression of oxytocin receptor or vasopressin receptor 1A. And in humans, he did a study where, people would rate kind of their success in lifelong pair bonding, if you will, and how much they enjoyed being, in a couple versus how, basically a monogamy scale. And they correlated the increased expression of, oxytocin receptor based on this polymorphism to increase feelings of monogamy. It's, it's a correlation, but it's another piece of the evidence that is used to suggest that really upregulation of oxytocin receptors is, a mechanism for this pair bonding. That being said, I think that in humans, the prairie vole versus mouse studies are really nice because it allows us to sort of tease apart what I think are two separate functions, this magnocellular versus parvocellular. But in humans, I think we certainly have both, and that, the parvocellular functions are still existent in the nucleus accumbens, but that it's just the release mechanism that determines whether it becomes, mad love or platonic love.
We should probably stop at that point.